Ladies and gentlemen, thank you for joining us for today's webinar, In Vitro Human Primary Cell-Based Screening Assays for Vaccine Adjuvant Development, presented by AllCells. My name is Qing Chen, Senior Applications Scientist at AllCells, and I will be moderating today's presentation. Before we get started, please be aware that everyone has joined today's program are automatically muted. And in order to participate in question and answer session immediately following the presentation, please enter your questions at any time using the GoToWebinar prompt at the right of your screen, and we will address them at the conclusion of the webinar. Today, it is my great pleasure to introduce our presenter, Dr. Erin Harberts from University of Maryland. Erin has a PhD in molecular microbiology and immunology from University of Maryland, Baltimore. She is currently working as a postdoc at Professor Robert Ernst's lab at the University of Maryland. Both Erin and Professor Ernst are participating in our webinar today. Please welcome Dr. Erin Harberts.
Thank you, Qing, very much for the introduction. Today I'm going to present some of our work that we've done in collaboration with MedImmune on developing rationally designed lipid A molecules for use as an adjuvant. Here's an outline of the talk I'll be giving today. I'm going to start by introducing vaccine components and the use of TLR4 agonists as adjuvants. And then I'm going to talk about the method that we have been using, bacteria enzyme combinatorial chemistry, which we've been calling BECC. And that's how we are going to make these novel lipid A TLR4 agonists. And then I will talk about some of our in vitro data using cell lines, and finally I will move into primary cell culture data, which is looking at the release of cytokines and also the upregulation of co-stimulatory markers. So to get us going, I want to talk about vaccines. Current vaccines have three main components. They have a delivery system, an antigen, and an immune potentiator, or an adjuvant. And we're not going to be focusing on the delivery systems today or the antigens. We are going to be focusing on an adjuvant. Historically, alum was the only licensed molecule for use as an adjuvant in human vaccines. And it has some good things about it, and it also has some pitfalls. It tends to skew the T helper response that's initiated towards a Th2 response, which is not appropriate for all antigens that you would want to vaccinate against. So more recently, the TLR signaling pathway has been invoked to initiate a more authentic immune response, and the engineering of TLR agonists to propagate this immune response is a hot topic of research, which I'm going to be talking about today. So when a B cell makes antibodies, you can see here that if it has Th2 help, it produces a strong allergic immune type response, which is not necessarily desirable for a vaccine model. Alternatively, if there is Th1 help or Th17 help, you boost more of a cellular immune response, and it will provide more appropriate lasting immunity for many of the pathogens that we would like to vaccinate against. So our goal in this project is to generate a panel of TLR4 agonist molecules that drive a Th1 and or a Th17 response. And these molecules must also not have endotoxin effects. They must not be pathogenic to the humans that they're going to be injected into. Now, why do we want to use the TLR receptor to propagate this immune response? Well, the TLR4 receptor is present at homeostatic levels on dendritic cells, which are important antigen-presenting cells when the propagation of an immune response. And you can see that the TLR4 receptor is present on both conventional myeloid dendritic cells and also the dermal resident BC, which are going to be very important for the site of inoculation of a vaccine. Now let me introduce you a little bit to the TLR4 signaling pathway. As you can see pictured here, in order for the immune response to be activated, the TLR4 signaling molecules must form a dimer in the membrane. Now, they're not pictured here, but this dimer also requires some accessory proteins that are membrane-associated, among them MD2 and CD14, which is the high-affinity receptor for LPS. MD2 and CD14 are responsible for presenting the LPS molecule to the TLR4. And depending on the specific structure of the LPS molecule and the way that it's presented to the TLR4, differential immune responses can be stimulated. And part of the differential stimulation of the TLR4 pathway is the fact that multiple transcription factors can be activated for the immune response that's propagating downstream of the signaling cascade. Today I'm going to be focusing on NF-kappa B as the transcription factor for the immune response we're going to be looking for. Here is a picture just to give you some perspective as to where the LPS is coming from. LPS is a main component of the outer membrane of gram-negative bacteria. And as you can see in the pull-out picture of the gram-negative membrane, the LPS molecule has three main components. It has an O polysaccharide, it has a core, and then it has the lipid A component, which has acyl chains on the bottom of it that anchor that LPS molecule into the membrane. Now, depending on which species the LPS is made in, which gram-negative bacterial species, that lipid A can have differential structure in the number and location and type of acyl chains that are added to the core polysaccharide. And depending on which species the lipid A is made in, as I said before, these acyl chains can differ. And the structure of the lipid A depends, the structure of the lipid A makes the decision as to what kind of immune response strength is propagated. On the left, you can see that E. coli has strong agonistic properties, and it is pathogenic to most humans that get that lipid A. And it results in sepsis and other bad things for humans. So we don't really want to use this lipid A structure. And then pictured here are three different lipid A structures that are made by Pseudomonas aeruginosa, specifically in the CF lung. You have the hexa, hepta, and penta-acylated version of lipid A. And as you remove those acyl chains, the immune stimulatory capacity of the lipid A decreases. And then on the other end of the spectrum, you have Yersinia pestis, which is a tetra-acylated lipid A. And that provides a nice backbone for us to make modifications to. As you can see, it's relatively immunologically silent. It doesn't cause a strong immune response to be propagated. And it also has a lot of spots open for us to add acyl chains to, which we can then subsequently trim and modify to tailor the immune response that we're propagating using our lipid A. And so we're going to be using Yersinia pestis, that tetra-acylated lipid A, in our system for the data I'm presenting today. And we use this technique called bacteria enzyme combinatorial chemistry, or BECC. And for this method, we express different lipid A modifying enzymes in trans in whatever workhorse bacteria we choose. In this case, we're using Yersinia pestis. And then we make that bacteria make the lipid A for us. It naturally will make the lipid A in large quantities. We can scale it up to a large batch. And then from a bacterial pellet from that culture, we can extract the LPS that's been produced. And then we can further chemically modify it to extract pure lipid A, which is a great TLR4 agonist, as I've mentioned before. And then we can take that LPS and lipid A and we can subject it to biochemical analysis, both mass spectrometry and GC, to confirm that the structure that we predicted would be made is actually being made. And then, once we confirm the structure, we can put that LPS and lipid A onto cells and also into live animals to screen them, and hopefully, at the end, we have identified a TLR4 ligand that has adjuvant potential. So that leads me to our central hypothesis, which is that using BECC, lipid A structure can be designed and altered to elicit an immune response that's desirable for use as an adjuvant. Because we can make so many of these molecules, and we have made so many of them, we couldn't possibly screen them all in human primary cells, although we would like to. So we created this screening pipeline to enable us to winnow down molecules to the most likely candidates for use as an adjuvant. We start by using immortalized cell lines, both the HEC blue and PHP1, which is a human monocytic cell line. The HEC blues are very special because they have a reporter underneath of the NF-kappa B promoter. So when we stimulate the HEC blues, we can take the supernatants off and very quickly and easily quantify the level of immune stimulation that has come from NF-kappa B. So that's a nice quick way to exclude or include molecules. And then once we go through our cell lines, we move into mouse primary cells, specifically mouse monocytes. And from those cultures, we take the supernatants and we read out the cytokine release using a Luminex assay. And then finally, once we've identified really well-characterized candidates for use as an adjuvant, we move into human primary cells, which is the focus of what I'm going to talk about today. We use human primary PBMCs, and similarly to the mouse splenocytes, we read the cytokine release using a Luminex assay. And then we also take human monocyte-derived dendritic cells, and we look for the capacity of our molecules to upregulate co-stimulatory markers on those dendritic cells, which is very exciting. And along the way, we are excluding molecules that generate both very high and very low inflammatory responses, because if the response is too high, it will be pathogenic to the human that we will inoculate with our adjuvant. And if it's not strong enough, then it won't potentiate an immune response that will provide lasting immunity to the recipient. Here is the screening setup that we have when we do our cell culture screens. We always test our lipid A molecules over 5 logs of dilution because they have a very big dynamic range for immune stimulatory capacity, and we want to make sure that we can hit that dynamic range. And also on every plate we include E. coli as a positive control, and then a known adjuvant comparator. In this case, we are going to be focusing on GLA or FAD. And then from that cell culture, we can take the supernatants and either do our reporter readout or cytokine detection from those cells. So here's an example of some of the reporter cell line data that we have generated. As you can see in this graph, E. coli, which is pictured in red, reaches its maximal stimulation level at a very low concentration. That is to be expected, and it's also indicative of pathogenesis for this molecule. And then you can see that the blue and orange lines, molecules A and D, have shifted the point at which they reach their maximal stimulatory capacity by about 2 logs, which is good for us. That's one of the things we look for because we can dilute them down and they will eventually reach a low or no stimulatory level. And these ranges are physiologic for injection. And then you have an example down here of molecule C, which is pictured in pink, which falls into the category of a reduced maximal stimulatory capacity molecule. These are also very exciting because no matter what dose we give it at, it will never reach the same pathogenic level as E. coli. And then in the bottom third, below the bottom dotted line, you have molecules that are very low have very low immunostimulatory capacity, and that we will winnow down out of the screening process. So now I'm going to move into some of the cytokine secretion data that we have generated. As I mentioned before, we first start with mouse splenocytes, and we have tested many different cytokines, and our Luminex panel is much larger than this, but these are the 8 cytokines or 6 cytokines that really give us usable data. So we have IL-8 and MIP-1 beta as chemokines, and those are responsible for recruiting immune cells to the site of injection. And you have IL-1 beta and TNF-alpha as markers of inflammation. And most importantly, we have IL-10 and IL-6, which are responsible for propagating and fueling the T helper response towards the differential T cell responses that we are looking for. Here's some of our mouse data. You have pictured on this one, in these graphs, you have E. coli in blue, and then you have SAD, which is a known adjuvant, in purple. And on each axis, you can see that E. coli stimulates the most compared to the other molecules that have been measured, which is to be expected. Again, E. coli's lipid A is extremely immune stimulatory, and it's pathogenic in humans. So we want to make sure that our molecules that we're moving forward are less than E. coli, but we also want them to be a little bit more than SAD. So we want to be a little bit better than what's already currently on the market, because we're always trying to evolve the technology. And you can see on the black 6 graph, specifically for IL-10, which propagates those Tregs and other differential T cell populations, that our adjuvants produce a lot more IL-10 than FAD, but again, less than E. coli, which is very exciting. Another exciting point about this data is that not only do we see that difference in black 6s, but we also see them in BALB-Cs. These mouse strains have very different genetic backgrounds. Black 6s are known to skew towards the Th1 response, and BALB-Cs are known to skew towards the Th2 response. So it's very exciting that we can see similar cytokine secretion in both of these genetically diverse mouse backgrounds. And it provides us a little bit of evidence that perhaps our molecules might have use in humans, which again are very genetically diverse. So then we wanted to move into human primary cells. We needed some PBMCs, and AllCells was recommended to us by collaborators at MedImmune to buy these PBMCs. And we decided to use them because they had high-quality cells and enough donors for us to use. So today I'm going to use these PBMCs, and I will stimulate them similarly to the mouse monocytes. And here's what that data looks like from the human PBMCs. I have pictured 3 donors, and as you can see, just looking at the graph, each donor has a very different immune stimulatory profile, which again is to be expected because humans are genetically diverse. You can see that donor 1 and donor 3 tend to make more IL-8, whereas donor 2 makes more IL-10. And although there are some differences among donors, overall, our immune molecules again stimulate more cytokine secretion than SAD, but less than E. coli, which is very exciting. It also highlights the fact that we need to screen this through many different human samples to make sure that we're having the response that we want to see. So then we wanted to move into using dendritic cells because while PBMCs give us a good snapshot, dendritic cells are the actual antigen-presenting cells which are going to propagate the immune response that we are looking for. And so we wanted to continue with this process, but we had so many molecules that we needed to contract it out, and AllCells was really responsive, and so we decided to work with them to continue for our dendritic cell assays. And here we have a picture just to orient you as to what the co-stimulatory markers on the DCs are actually doing. In the middle, you have MHC, which is presenting antigen to the T cell receptor. And then on the side, you have these co-stimulatory molecules, which are necessary for the T cell to be licensed to go ahead and propagate the immune response. So without these co-stimulatory markers on the antigen-presenting cells, an effective immune response will not be made. So we wanted to measure the ability of our molecules to upregulate these co-stimulatory molecules on human dendritic cells. And we did that by coming up with this workflow. Here we started with 4 healthy donors, and then we isolated the PBMC from them, and we selected for CD14 positive monocytes, and then we matured those CD14 positive monocytes for 7 days with GM-CSF and IL-4 to create monocyte-derived dendritic cells. And then once we had a pure monocyte-derived dendritic cell population, we put our molecules onto it and then we used FACS to read out for the upregulation of those co-stimulatory markers. And as I mentioned before, we had so many, so we contracted out, and AllCells did this for us, but we helped them come up with the protocol. And here is the data that came back at us when we sent away our molecules. It's a lot of data, and it's pretty informative. Here I have pictured 4 different co-stimulatory markers, CD83, CD80, CD40, and CD86. And you can see that along with our compounds, we ran GLA, which again is our known adjuvant comparator. We have E. coli LPS as our positive control, and then CpG as a differential TLR agonist, so another control. And overall, among the 4 donors tested, we see that our compounds have a middling stimulation, which is what we want, more than GLA and less than E. coli. So this was very exciting to see that we can actually upregulate co-stimulatory markers on DCs with our novel TLR4 ligand. And then not only did we do them at one concentration, which is pictured on this slide, we also did a dose response at the same time. So we tested 3 different log concentrations of our molecules and their ability to upregulate those co-stims. Here is one co-stim, CD83, which is pictured. And while there is lots of variability among donors, you can see that the percent positive ranges pretty wide between donor 1, donor 2, donor 3, and donor 4. But it was really exciting to see that at the maximal dose that we tested, our molecules generally parsed out at in the same order as for their immune stimulatory capacity, no matter which donor was tested. So this is very exciting for us, and we think that we have some molecules in hand that will be useful as a vaccine adjuvant. And then finally, I want to picture here that you can also pull out the MSI data from all of these experiments to be able to elucidate a little bit further some of the nuanced activation levels of these dendritic cells. And if you pull out 3 different markers and you do the 4 donors for the MSI, again, you can see that our molecules stimulate just a little more than FAD in most cases, but definitely less than E. coli and definitely more than untreated cells. So, so far, it seems to be working, and we're pretty excited about this data. In conclusion, we have created these rationally designed lipid A molecules using BECC as a method to engineer the structures that we want and that we think will signal in a specific way through TLR4. We then selected down all of the molecules we made for ones that might be useful as an adjuvant. And along the way, we've compared it to known adjuvants and molecules that are under investigation for use as adjuvants. And I'm not going to talk about it today, but we have preliminary data in well-described virus and bacterial protection models to show that our molecules are indeed able to provide protection better than an unadjuvanted antigen, which is pretty exciting. It's preliminary, but we're excited to move forward with this project. And with that, I would like to acknowledge everyone who helped with this project, specifically Kelsey Gregg, who has been side by side with me on all of these experiments, and definitely our collaborators at MedImmune who ran all the Luminex, and the AllCells collaborators who ran the dendritic cells for us and provided us with the human primary PBMCs. So with that, I would like to hand it over back to Ching.
Thank you, Erin, for the great presentation. Well, it certainly taught us that primary cell-based assays provide more physiologically relevant data, but when we study the primary cell-based assays, we need to keep in mind it's always a range of response because of the donor-to-donor variation. So before we jump into the QA session, I would like to use a couple of minutes to summarize what AllCells offer. And what does AllCells offer? AllCells offers fresh tissue, including whole blood, bone marrow, local pack, mobilized local pack, and cord blood. And we also offer the isolated cells from fresh tissues. On top of that, in addition, we start to offer the cell-based assays, including phenotyping, cell expansion, and compound screening. For example, this DC maturation compound screening assay that Erin just presented. And we offer a wide selection of purified primary cells in fresh and frozen. Next slide. And in summary, AllCells has served the life science community as a reliable source of blood-derived primary cells and services for over 18 years. So our local lab is the clinical division of AllCells, providing the researchers immediate access to well-managed donor pool. We control the whole process from donor selection to cell isolation. And therefore, we have the freshest starting tissue, which ensures the highest quality and the consistency of the product. We are running a webinar attendee promotion at this point, and please follow up email for information on how to redeem the 20% off cultured cells, including dendritic cells, macrophage, and mesenchymal stem cells. So we will now begin answering any questions that you may have. As a reminder, please enter your questions in the GoToWebinar prompt at the right of your screen. And let's go to our first question. Well, first question is for Bob and Erin. Actually, this is really exciting research project. What are your next steps?
Great question. Our next steps are obviously to try our adjuvants in known protection vaccine models, which we are currently working on, and we have some data that it will work. We need to expand it to more physiologically relevant vaccine models, and ultimately the goal is obviously to be able to use our adjuvant with an antigen and create a vaccine for human use.
Oh, great. And I also have a question just from a customer perspective. What's your experience with working with AllCells?
Yeah, we had a really good experience with AllCells, which is what gave us the want to run all of our dendritic cell assays with them. They have a good pool of cells. Every time I saw them, they are pretty healthy, and they have a large donor pool and a lot of vials per donor that are available, which is good for us as researchers because we want to be able to go back to the same donor. And they were very helpful in coming up with the protocols that we use for our dendritic cell assays. They ran them to our specs, and I'm sure that they would be willing to run them to other people's specs too. It's kind of like working with a lab mate that you have. So it was great.
Well, thank you for the nice feedback. And if there are no more questions, that concludes our webinar for the day. Today's webinar will be posted on our website. And for the product or services related questions, please contact sales@allcells.com. And on behalf of Erin, Professor Ernst, and the AllCells team, I would like to thank you for your time and interest today. We look forward to host more educational presentations in the future. Thank you. Have a nice day.